Go ahead.
Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Professor Zygmunt Derewenda from the University of Virginia, who will talk about hydrogen bonds and protein secondary structure. Over to you, Zygmunt, please.
Okay, I'm sorry about the technical issues. I really don't know where the problem is. It's of course great fun and pleasure to be with you this morning or this afternoon, depending on where you are. I'm speaking from the University of Virginia. You can see the historic campus here, designed and built by Thomas Jefferson. By English standard, this is very young, but by our standards, 1817, I believe, quite an old university. All right, let's move on. So the idea, and right, next slide, you can see, I'm assuming. Okay, very good. So I'm going to start with a brief introduction, and then I will try. My understanding is that the interested group are mathematicians and others interested in patterns of networks of hydrogen bonds. And so let me give you a brief introduction, which will then help me move on with the topic at hand. So proteins are polymers of 20 amino acids and are linked by identical peptide units, which was discovered early on in the 20th century by Emil Fischer. And you can see here the nature of this so-called dehydration synthesis, where a carboxyl group and an amino group are joined by a peptide bond while a water molecule is being expelled. Very early on in the 1930s, some of the first crystallographic studies of organic compounds showed that diglycine, which is basically the simplest possible compound that includes 2 amino acids, that the atoms that form this peptide bond that I'm showing you here, these are in fact 6 atoms, they all lie in one plane. This was an interesting observation, but at the time there wasn't quite an explanation why that happens until Linus Pauling, perhaps the greatest chemist of all times, but certainly 20th century, invoked the principle of so-called resonance. In other words, he said that the types of bonds that link these atoms fluctuate, and this fluctuation imposes planarity on the arrangement of the peptide bond. You can see here double bond between carbon and the nitrogen, which then migrates into the carbon-oxygen. And because these 2 structures switch between them very quickly, this entire ensemble is in one plane. The other consequence of that is that partial charges develop on these atoms, specifically the oxygen and the NH group. And you can see the table on the right hand that shows the partial structures on the peptide group. Now, the formation of peptide bond is actually one of the most complicated phenomena in biology, and in cells, it is catalyzed by this huge nanomachine that is still under intense study. Also, one of my colleagues here in the department is involved, Ahmad Joma, in these studies. We're not going to go there and talk about the big machinery. We just want to focus on the peptide bond itself a little bit more because it is critical to the discussion of hydrogen bonds and protein secondary structures. So now that we know that these 6 atoms, as you can see here, between 2 amino acids are in one plane, the question is it this configuration known as cis, where the 2 residues are on one side and you have the oxygen and hydrogen here? Or maybe if we just flip around the central bond, we get the transpeptide, which is kind of more straight, more elegant, and also there is no clash between these residues right there at the top. So in reality, our first lesson here is that, or rather second lesson, this is a flat ensemble, and also in most cases, this will be a trans peptide in the vast majority of cases. There is one regular exception, and that is with proline. Proline is one single amino acid that is cyclic and does not have a free hydrogen on the NH group. Instead, there is a pentameric cyclic structure. In that case, it actually doesn't matter for steric purposes if there is a clash on one side or on the other. The two configurations are basically kind of seen almost at equal frequencies. The most important lesson that will now carry us into the hydrogen bonds is the fact that these charges on the peptide group, in fact, generate a dipole moment across the peptide group. The dipole moment is a measure of the polarity. You have an ensemble which is charged negatively on one side and positively on the other. And in fact, the dipole moment of the peptide bond is twice that of water molecule, and water molecule itself is quite polar. So the consequence of this is that you have here a situation, if you remember from Steve Shiner's talk, which is very favorable for the formation of the hydrogen bonds, because here you have a hydrogen attached to the nitrogen, which may act as a donor, and then you have the oxygen, which is polarized and negative, which can be an acceptor. And interestingly enough, even though proteins are a product of evolutionary combinatorial chemistry, there is this peptide bond which repeats itself, identical, 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 but it actually does impose a structure, as we will see on the next slide. So now we understand that the polypeptide chain is basically a link of these main chain groups, which are planar, and there are only 2 degrees of freedom between two residues. There is a twist around this bond, which is known as phi, and a twist around this bond, which is known as psi. And so we then move to another residue, and we have again these 2 angles, and this is how we can define what is known as the secondary structure in proteins. So the secondary structure is the actual configuration of these residues with respect to one another, while the primary structure, of course, is the actual sequence of amino acids in the polypeptide chain. Now, if we take those 2 parameters, phi and psi, and chart them, make a graph, and use the available knowledge, the database of proteins as they exist in nature, we show it on the right-hand side, then you will see that we have specific areas of this chart that are populated and many that are forbidden. Why are they forbidden? Because if you select that combination of conformational parameters, you end up clashing. Some atoms will clash into other atoms. And so those are really impossible in terms of steric collisions, in terms of energy. So you end up with 2 primary regions, alpha and beta, which we will soon see are helical and sheets. And there are also other regions which I won't necessarily talk specifically about, but I want to give you an introduction of how hydrogen bonds define especially those 2 areas. So the discovery of secondary structure elements, which are repeated in protein structures, is credited to Linus Pauling again. Linus Pauling has accomplished many things, and many of his papers contain very dramatic mistakes, but he is not remembered for the mistakes. He's remembered rather for the things that appear to be correct. So on the left-hand side, you see his first suggestion of a helix, I won't go into the anecdote how that happened, but you can see 1951 are the papers which revolutionized essentially protein chemistry. That was even before any structure became known. He hypothesized, modeled this, except that the helix on the left-hand side is actually incorrect because it is left-handed instead of right-handed, and the one on the right that he envisioned does not exist in nature. So this paper is 25% correct. We will go into the exact details. In this modeling, however, a very important role was played by perceived hydrogen bonding between successive peptide groups. So what I just told you about, the peptide group at one end can be a donor, at the other acceptor, and how they interact with each other then defines the secondary structure element. On the right-hand side are figures from another paper that came out the same year, extremely productive year for Linus Pauling, in which you see the principle of anti-parallel and parallel beta sheets. Again, note that Linus Pauling placed oxygens and the NH groups next to one another, and he invoked the presence of hydrogen bonds based simply on modeling and the distances in the geometry. Now, Steve Shiner last time during the previous lecture explained to you the question of cohesive energy, attraction. Here, looking at the structure, we basically infer, we assume that if these two groups are close together, it must be a bond, it must be cohesive. But we don't have a proof. We basically have proof from organic chemistry that such interactions are cohesive, but in proteins, these models were just assumed to be stabilized by those hydrogen bonds. Now about helices and what hydrogen bonds stabilize. In fact, we've learned after many protein structures were solved that there are three principal types of helices. It's not as simple. In the upper part of the diagram, you see a diagram of a polypeptide chain. And so if the oxygen, forget about the first one, but if the oxygen interacts with the hydrogen on the third residue, you get the so-called 310 helix. And then if it's the next one, then it's an alpha helix, and then it's a pi helix. The notation is confusing, I think for historical reasons, because actually that's not how it arose. Nevertheless, I think someone decided that because there is no Greek letter prior to alpha, alpha is the first one, then there was nothing to, no name to attach to a simpler helix. So they came up with 310. What does 310 mean? It means that the residues connected by hydrogen bonds, it's 1 to 3, and the number of atoms in the loop connecting the two atoms in hydrogen bond is 10. There are 10 atoms. You can count them in here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? And so that became a 310 helix. If we follow this terminology, which will be, I think, very easier, the alpha helix becomes a 4-3 helix. So it's a slightly looser helix connecting residues 1 to 4 with 13 atoms in this loop. The pi helix, which was not predicted, none of these 310 or pi helices were predicted by Pauling, but the pi helix is even looser in that it connects every fifth residue by hydrogen bond, and there are 16 atoms in that loop. Please note, when you look down the axis, here I'm showing on the left lower corner, this is how the 310 helix looks, very simple, like 120-degree steps. The pi helix is more complex because, of course, the pitch is not integral, and the same is true for pi helix, which, as you can see, has a significantly larger radius. Now, as these helices form, those interactions mediated by hydrogen bonds are enhanced by the fact that the dipole moments, which are shown on the right here, the electrostatic potential, stacks on top of one another. So in fact, those dipoles reinforce one another. And in terms of electrostatics, the helix becomes more stable due to those interactions. You can see the partial positive charges in a helix lining up against partial negative charges, and that is what stabilizes the structure. So to a large extent, as far as the alpha helix, Linus Pauling was correct. What about the beta sheets? On the left-hand side, you see what is known the anti-parallel. In other words, each strand goes in the opposite direction as the arrows show. And that places conveniently the carbonyl groups and the NH groups opposing each other. And so you have a pair of hydrogen bonds here, while on the other side, you have a complementary set of hydrogen bonds that, as you can see, are displaced in a relationship to the first pair of oligopeptides. When you look from the side, because the carbon atoms are tetrahedral in these, of course, amino acids, you end up with a sort of a pleated arrangement with the side chains of amino acids pointing either on one face of the beta sheet or on the other. Now, anti-parallel, it doesn't need to be that way because you can switch the direction of the middle strand and get a fully parallel beta sheet, which will also be stabilized by hydrogen bonds, as you can see here in the center, although those hydrogen bonds, in terms of the stereochemistry, look somewhat different, and especially the angles deviate from this sort of parallel arrangement that you can see on the left-hand side. Looking from the side, you would never know that one is parallel, the other is anti-parallel, because the same principle holds that the residues fit on both faces. Now, the model was nice, but the reality is much more complex. And I have to say that I have not found a paper that would explore the relationship between hydrogen bond precise stereochemistry and the fact that these beta sheets are always twisted. You can see on the right-hand side there are four strands that are shown, symbolized by four fingers of the hand. And if you hold four fingers, you will understand how this twist occurs in nature. And it, of course, impacts the hydrogen bond network in between, whether that is a driving force for the twist or the hydrophobic interactions. I think it's rather the hydrophobic interactions, and the hydrogen bonds adapt in a very specific way. Nevertheless, it's the twisted orientation that is common, dominant, and not the flat one, which was originally modeled. Now we're going to get a slightly more complicated issue, and we want to jump into the details of those hydrogen bonds. So far, what I said was that the oxygen comes within a certain distance of hydrogen, as was explained by Steve Shiner a couple of weeks ago. And we just say there's a hydrogen bond. But in fact, the situation is way more complicated. Let's look at the structure of atoms. Atoms have nuclei surrounded by electrons, and electrons come in pairs. It's just the way it is. And those pairs occupy what is referred to as orbitals. And depending on how many electrons you have and what are the bonds in between them, those orbitals can switch and or even come together and form hybrids. So the outer shell of these elements that we're interested in, and this is nitrogen and carbon, remember the NH is the donor, oxygen is the acceptor, in fact are in the so-called sp2 hybridization, which means that there are three pairs of electrons that are in one single plane. That is a part of explanation of why this entire group, peptide group, is planar from the point of view of quantum chemistry. Those orbitals force the planar arrangement. Now, when you look at the carbonyl or CO bond, it is complicated in that it involves the s and p orbitals, but suffice it to say, jumping straight to the important part, is that from oxygen, you have these two electron pairs, free, coming out at approximately 120-degree angle. Now, if you remember what Steve Shiner told us about hydrogen bonds, is that the proton from the hydrogen approaches the free electron pair of the acceptor. So the oxygen of the carbonyl group of the peptide bond actually has a potential to form two hydrogen bonds, not one, but two. All the other models that I showed you earlier were all based on the concept that we have a single hydrogen bond. But that's oversimplification. It gets even more complicated when you look at the diagram at the bottom. On the left-hand side is the simplistic view, which was used by Pauling. In the center is, you know, a bifurcated bond, which is possible because the oxygen has two electron pairs. Each one of them can form a separate hydrogen bond. And then you have the situation where one hydrogen from the NH group, in fact, can interact with two oxygens on separate carbonyl groups. So the stereochemistry and the pattern of hydrogen bonds mediated by the peptide groups is very complex and occasionally even difficult to describe in a single uniform way. This has very important consequences, because the next question I'm going to ask is, all right, so if the oxygen has the potential to make two hydrogen bonds, why have we been talking about only one? That doesn't make sense from the point of view of chemistry. Well, when you look at high resolution structures, and here is a piece of an alpha helix shown together with the electron density at relatively high resolution. This is a carbonyl that makes a hydrogen bond with the fourth residue up in an alpha helix. And you can see that this involves one orbital, sp2 orbital, whereas the other one, it's potentially saturated by a water molecule that is sitting right next to the helix. It may be also a residue from an adjacent helix. It may be something else, but those carbonyl groups tend to have always a second partner, which also impacts on the overall structure. In beta sheets, it in fact gets more complicated, because when you look at the structures carefully, and I'm showing an example here in the upper left-hand corner, you will see that the carbonyl group is in fact in steric proximity of two groups and potential donors. Let's look at this one. It is, you have here the NH group, but here you have a proton that is attached, a hydrogen attached to a C alpha carbon. In other words, the alpha carbon central atom of an amino acid. On the right-hand side, you have what is known as difference electron density, which actually shows the presence of those two protons and the distances. So here, what I'm suggesting, what I'm actually telling you is that there is another class of hydrogen bonds that were not predicted by Pauling and that have, but they do come in play in beta sheets. And here at the bottom, a little bit of self-promotion. For a while, we held the record of the highest resolution and most. Anyway, here you have electron density for hydrogens. This one is attached to an alpha carbon. And here you have the, it says again that my internet connection is unstable, but it disappeared. Okay, so you can see every single is attached to a C alpha. Now, a little bit of history. I love science history. In fact, even though Linus Pauling is being proposed as early as 1933 by William Asbury, and then in more even precise way in 1937 by Maurice Huggins. So the notion that hydrogen bonds will stabilize the chains goes back way before Linus Pauling put specific coordinates. The ideas were already in 1942, Maurice Huggins was looking at models of structures, including collagen, and was suggesting the possibility of the CH groups acting as donors in these hydrogen bonds. This was proven in 1996, so half a century later, by Helen Berman, and it was shown at high resolution that indeed such interactions occur. Now, a bit of self-promotion. I'm terribly sorry, but you know I can't help myself. So in fact, in 1995, we were the first group to discover the presence of these double hydrogen bonds in beta sheets and also the presence of CHO bonds generally in proteins. And in fact, it did fill in a significant gap in our understanding of secondary structure. But we were, of course, able to show proximity. And when we did that particular, wrote a particular paper with Ula, who's listening to this too, we based this, believe it or not, at high resolution. That was the limited number of protein structures available in 1995. But we were still able to statistically show that those interactions may be important. Now, 10 years later, Steve Shiner, whom you listened to, quantum chemical calculations, that the energies of these interactions are also very important. It's just not serendipitous proximity, but in fact, energetically, those interactions are very important. And in some cases, the CHO approaches in terms of energies the classical canonical hydrogen bond that involves the NH group. Well, that ends my self-promotion. And then let me give you a few more details about how to illustrate how complicated these networks are. Again, we continue to speak about hydrogen bonds mediated by the peptide groups arranged in the secondary structure motifs. So one of the important features, important properties of these bonds is that they can stabilize bundles of helices, which occur frequently in membrane proteins. And here, I believe, an Academy member who looked at the interactions in a variety of proteins, very important proteins that cross the cell membrane, and discovered that, as I was telling you earlier, there is the second orbital which interacts with the CH from another in those bundles together. So one more example of the complexity. And now let's come to another type of secondary structure, specifically known as turns. Well, you cannot have a helix running infinitely into space. You cannot have a chain must turn. And interestingly, those turns are also governed by hydrogen bonds between nearby amino acids. In the upper right corner, I'm showing you what is known as the beta turn and the hydrogen bond, which is characteristic for this type of a hydrogen bond that goes from I to I+1, I+2, I+3. And this is a very rigid arrangement with a flat peptide right here. And this is how the polypeptide chain can turn. There are, in fact, varieties depending on the exact stereochemistry. And we will now have one example where a side chain plays a part, and I'm coming to the discussion of how side chains are involved. Here you have not a beta turn, but an Essex turn. They kind of look similar, but I want you to note one very important difference. Here, you have a main chain that meanders through this turn. This carbonyl and this NH group both come from the main chain of the protein. Here, you have a switch. The main chain, in fact, goes this way, whereas the group that comes in and stabilizes this switch or turn comes from the side chain. This is asparagine, and the carbonyl group comes from asparagine's side chain. So you have a turn that changes the path of the polypeptide chain, but it is mediated by a peptide to side chain bond. Now, speaking of side chains, so far, I've been focusing on main chain interactions, which are really the defining features that not only characterize the secondary structure, but because of their involvement in turns and how the different secondary structures come together, that defines, of course, the tertiary structures. There are several amino acids which are charged. The positive charges are carried by arginine, histidine, and lysine. And we have two negatively charged amino acids, and that is aspartic acid and glutamic acid. And those side chains can form very strong bonds that are known as charge assisted. I think we mentioned that when Steve was giving his talk, because in addition to sharing a proton, these hydrogen bonds have very, very strong electrostatic attraction. And someone last time asked the question about salt bridges. Well, salt bridges are so if you have arginine next to glutamic acid, this will be not a classical hydrogen bond, but it is going to be a salt bridge. That's how this is called. You also have amino acids with polar uncharged side chains, such as serine, threonine, asparagine, and glutamine. No charge, but they're, of course, capable of forming hydrogen bonds through their OH groups. There is also tyrosine in that category, which somehow got cut out of here. And I'm going to leave it at this and just show you examples. We started a little bit late, so I assume I have about a few more minutes, but I want to give you examples how side chains interact with main chain to deliver additional features stabilizing tertiary structure. You've seen the ASX turn already. Here is an aspartic acid, which interacts with a main chain NH group and allows the switch in this polypeptide chain while maintaining this hydrogen bond. In a similar way, a serine here, this is a side chain hydroxyl group, can interact with NH and basically confer the same stereochemistry. In places where helices, you have residues that cap the peptide groups, either carbonyls or NH groups, in order to keep the hydrogen bonding potential saturated. So here you have an example, two examples of histidine coming at the end of a helix. Now, this NH group would be left alone, unpaired, unhappy, lonely, and so this side chain of histidine comes in and saturates its potential, makes it happy, while the main chain can meander in a different direction. The same is true shown here, and the histidine, in fact, utilizes its nitrogen, which can serve as an acceptor, not the one which can serve as a donor. Now, maybe you can see this very clearly, but this is actually a helix. This is the right-handed alpha helix here. Again, electron density at high resolution, which clearly shows how the asparagine here engages the carbonyl. So now it's the carbonyl that is being capped from the top, aside from the main chain nitrogen, and then the main chain meanders away while this structure stabilizes what is known as the cap. Finally, I would like to show you something that we have recently worked on together with Steve Shiner, and we brought in crystallography and analysis database, and this was also a collaborative work with Vladek Miner's lab. The three of us came together, and we looked at a very interesting feature of one specific amino acid, which is tryptophan, and it has one polarized CH group right here, which is being used in the same way as you have seen the example with histidine or with ASX to cap certain carbonyls in very unique pieces of secondary structure. This was published recently in Acta Crystallographica. So you can see here that you have turns that are basically to the next residue. You have a residue that is the residues that are separated by one. And interestingly, this is almost similar to the beta turn. You can see the main chain taking a kink and meandering in the other direction, while the tryptophan maintains through this hydrogen bond the saturation of the carbonyl. Other examples in the same group, and here you have a turn that includes one extra residue, and you have different configurations, including a double hydrogen bond, one from main chain NH group, one from side chain CH group. And so what I'm trying to say is that we have these general patterns, the alpha helices, 310 helices, pi helices, very regular hydrogen bonds, but we have to remember that the hydrogen bonding network is more intricate, involves almost always 2 bonds per carbonyl, and those comes from water or side chains or some other elements. And then when you look at the turns, it really gets very complex because you have both main chain, side chain, and side chain interactions mediated by hydrogen bonds that stabilize this feature that, when put together, give you a full tertiary structure of a protein. And I believe I'm done. Thank you very much. I hope that, you know, I introduced the subject a little bit to you, and of course, I'm happy to answer questions.
Thank you very much, Zygmunt. Thanks a lot. Please thank Zygmunt for the beautiful presentation. Physically or virtually.
Thank you.
I'll stop